Hi there. In this video, we're going to explore program management and the translational framework on Pluto, and we'll do this in the context of an example program on rheumatoid arthritis. Pluto is infrastructure for translational science, and the highest level of organization on Pluto is programs. So you can track all of your programs using this Kanban style view and move them from one program to the next. And you can really easily just get a snapshot of program name, stage, modality, therapeutic area, target, and health. Going into one of these programs, on the left-hand side, you can see all the components that make up a program. Things like the framework, datasets, analysis, and targets. And many of these are things that exist, but they're often scattered across spreadsheets and Google Docs and emails and your local drive. And it becomes really difficult to find the data that you need. And often work starts getting duplicated and it's just not happening very quickly. So what this is meant to do is consolidate all of the data and reports and information that you need to run a program in one place so that it's collaborative and everyone can work on it in real time. And also what that allows Pluto to do is continuously update something called the Translational Alignment Framework. The translational framework consists of 42 criteria for translational program success, and these were developed by Pluto, and they span biology and disease discovery, target and therapeutic actionability, model and disease alignment, and mechanistic and functional confidence. These criteria are continuously updated based on what analyses you've run in Pluto, what documentation you brought into the platform, and ultimately they're also used to recommend next steps. So you can see for dimension 1 here of this program that 5 different criteria have partially been completed, 3 are missing, and these are the proposed next steps, which we can just click to go and analyze in Pluto. So it basically guides you through the process of completing your translational framework. Ultimately, these criteria are filled out by pulling in data and running analysis. So let's grab some data. Datasets in Pluto can be brought in as either CSV files or FASTQ files, or they can be grabbed from the Global Data Commons. The Global Data Commons allows you to import any GEOS dataset that you want just by inputting the GeoAccession ID, and that will be fetched automatically. Or you have access to 15,000 published datasets that are already available in Pluto. So in this rheumatoid arthritis program, we already have 2 datasets. I'm going to navigate into this dataset. You'll notice that this is a public dataset pulled from the GeoData Commons, but if this were an internal dataset, you could just upload the count CSV and the sample annotations file, and then you'd be ready to begin analysis. So you can click on Analyze this dataset, and that will take you to the Pluto canvas. The canvas is like a functional scientist whiteboard that updates in real time, like a Google Doc, so you can have multiple people on here running analyses simultaneously. You can add notes and really start to tell a scientific story. But all of the analyses that you're seeing here, like PCA, differential expression, gene set enrichment, summary plots, are generated using non-coding graphical modules. So you can just click to create a new analysis, select your data set, select the analysis type, and then any of the variables that were uploaded in your sample metadata are available to explore. So let's say that we want to actually look at a combination of condition and tissue, and then compare inflammatory arthritis peripheral blood to healthy peripheral blood. We can run that analysis, and then you'll see this nice volcano plot return to you, which you can edit to meet your needs. So you can change colors, for example. And then importantly, you can also explore lists of genes. So we could label an entire biomarker list, for example, JAK-STAT signaling. Or we may want to label individual genes by searching for them. In this case, what I actually want to do is to basically lasso some of the most significantly increased and decreased genes kind of at the top of the plot. And then I want to save those to an ongoing list. So I'm going to select existing list and add these genes to my RA targets list. You may also want to save specific plots to certain genes in that list as evidence. In that case, you can click to save a certain plot as evidence, select the target list, and select the gene of interest, and save. So then you can go to the targets in your program, select that target list, search for that target, and then you can see an AI summary of that target, the predicted 3D structure, and then the evidence that you saved from the canvas. Hopefully this gives you an idea of how analysis is run on Pluto, how that analysis is used to continuously update the TAF, and how that's used to move programs forward. Thanks for watching. Bye.